are happy to welcome Bruno Paiva for this new Red Talk. So Red Talks is a webinar series dedicated to open innovation in lymphoid neoplasm, organized by the Carnot Kalim Institute. Our goal is to bring together experts from academia and industry to discuss the latest breakthrough and challenges in the field. For those who don't know, the Carnot Kalim Institute is a French research consortium specializing in lymphoid hemopathy. They unite top-tier academic laboratories and clinical research centers to accelerate innovation and improve patient outcomes. So the talk today is entitled Myeloma Patient Follow-up System with MRD Algorithm from Dr. Bruno Paiva, and Jérôme will introduce Bruno.
Yeah, so hi everybody. So we are really happy to welcome Bruno today. So Dr. Bruno Paiva is a leading expert in multiple myeloma and minimal residual disease assessment, and as a co-director of the hemato-oncology program at the Universidad de Navarra. He has been at the forefront of developing advanced strategies for disease monitoring and patient follow-up, especially using multidimensional flow cytometry and also immunogenomics. And his work bridges clinical innovation and scientific rigor, particularly in the use of MRD algorithms to enhance precision medicine approaches in hematology. And thank you again, Bruno, to be there today.
Good afternoon to everyone. It is my great pleasure to be here among some French colleagues and to spend the next hour with you, sharing some of our results, also ideas, and hoping also to learn from you during the Q&A session that will follow the presentation. I will now start sharing my screen. And I would please ask to someone to confirm that you can all see my presentation. Yeah?
Just a minute.
Okay, so you can see my presentation. So if you see my presentation...
No.
Not
you
yet.
cannot?
No, we can't.
Oh.
It is starting. Yeah.
Yes.
But now you can.
Yeah.
And you still see my slide?
Yeah, perfect.
Okay. So if you are seeing the slide, then you realize that I changed the title of the presentation. I will talk about MRD, that's for sure, but I changed it into a more general title so that it can be of interest, hopefully, to everyone listening, regardless of your interest or work specifically in the field of MRD. These are my disclosures, and this is the outline of my presentation. I will first talk about what we believe could be biomarkers of consequential disease, and I will address what this means in a minute. Then the use of big data to try to answer some of the big questions out there in the field of myeloma, and then finally trying to push the limits, the boundaries of technology to improve patients' quality of life. In this slide, I summarize what is a personal conceptual hypothesis about the origin of monoclonal gammopathies, in which the expansion of oligoclonal B cells and plasma cells that often carry important genetic alterations such as translocations, hyperdiploidy, somatic mutations in oncogenes, with the subsequent detection of a monoclonal immunoglobulin that is as frequent as more than 4% in individuals aged over 50. This could be no more, no less than aging-associated alterations or maladaptive aging, or eventually something that we all know and frequently define as a monoclonal gammopathy of undetermined significance. And if we accept this as almost a natural finding and a consequence of aging, then we need something else that triggers consequential disease, malignancy, such as myeloma. And we believe that two potential triggers of consequential disease are circulating tumor cells and immune tape. Because of this, in the last decade, we had a special focus on circulating tumor cells. And we started by isolating tumor cells from the marrow and from the peripheral blood from the same patients, after which we performed DNA sequencing, whole exome sequencing, and we found that there were no unifying genetic events associated with tumor regress from the marrow. In other words, if you looked at the genetic alterations in CTCs, 80% or more were similar to that found in the bone marrow. And while these results are promising, positive, towards the concept of liquid biopsies, for me it was disappointing because to some extent, we could not explain the tumor regress from the marrow based on private genetic alterations. And then we went on and characterized transcriptional profiles, again, of patient-paired tumor cells from the marrow and the peripheral blood, carefully isolated using flow cytometry based on patient-specific phenotypes. And it couldn't be the other way around. We found highly similar transcriptional profiles because the genetic profile was very much similar. Only a few genes significantly differentially expressed. And interestingly, these few genes converged into one or two pathways that were suggestive that fully occupied and hypoxic bone marrow niches together with a pro-inflammatory tumor microenvironment were probably forcing myeloma cells to stop proliferating. The conditions were not there anymore for proliferation, and because of that, these cells were forced to recirculate in the blood, basically to seek, to find other bone marrow niches to continue growing. In other words, if this hypothesis was true, then the numbers of CTCs in the blood would be a surrogate not only of tumor burden, but also proliferation, niche occupancy, and definitely of disease dissemination, and therefore an exquisite prognostic factor. And in fact, more recently, many studies are showing that the higher the CTC numbers, the greater the risk of disease progression in myeloma. And to some extent, to test the hypothesis, that conceptual hypothesis about the progression of myeloma, the origin of consequential disease, 8 years ago, we initiated the Immunocell study. This was a large international multi-center study, by the way, with some of your French colleagues in Toulouse participating in the study, in which we enrolled slightly more than 300 patients with smoldering myeloma, any risk, untreated, that were to be followed in the peripheral blood every 6 months during a period of 3 years, and whenever possible in the bone marrow every year. And in these blood and marrow samples, we were using a next generation flow cytometry, or NGF, to look both at tumor cells, particularly CTCs, as well as for immune profiling. Briefly, because I will cover many different topics briefly, in this slide, we show how it is possible to create a new model to stratify the risk of patients with smoldering myeloma from the original 2020 model developed by the International Myeloma Working Group that relies on the concentration of the M protein, the serum free light chains, and the other 20 stands for the percentage of plasma cells in the marrow assessed by conventional microscopy. This is the model, and this is the risk of progression according to high, intermediate, and low risk. And in the right part of the slide, we have a new model that keeps the concentration of the M protein, the serum free light chain ratio, less or more than 20, and a new cutoff of 0.01, 0.015% circulating tumor cells. And as you can see, we replaced bone marrow plasma cells by CTCs, and the models are very much similar. This is similar risk stratification, but obviously a clear advantage of the model in the right part of the slide. And the clear advantage is that it relies on 3 minimally invasive biomarkers: 2 in the serum, M protein, free light chains, and the CTCs in the peripheral blood. And we are looking for minimally invasive assessment of tumor burden because this is where we want to go. We do not want to replace the bone marrow by peripheral blood. In my opinion, the marrow will remain a gold standard sample to assess patient's risk. Rather, we want to offer more. And what we want to offer is a possibility to stage patients with smoldering myeloma based on the marrow and in the peripheral blood tumor burden. And because it's minimally invasive, that means that every 3, 6, or 12 months, when the patient returns to the consult, you can continue recalibrating the patient risk by looking at the serum and the peripheral blood, and thereby determine stable versus evolving patterns, and thereby improve the prediction of the future of the patient, if he or she will progress or not into active disease. This is preliminary data, and by the way, only looking at CTCs without all the other parameters that are complementary. But instead of a single assessment, as in the previous slide, this is already leveraging on the kinetics of CTCs, almost 800 assessments that allowed us to stratify patients into 3 subgroups: those with sustained and detectable CTCs, virtually no risk of transformation. This is not smoldering myeloma. This is more MGUS-like disease or purely benign condition. And then 2 subgroups with detectable CTCs, but in green, stable levels every 6 months, very low risk of transformation. By contrast, evolving levels, significantly higher risk of transformation. We do believe that the kinetics of CTCs, as well as M protein, hemoglobin, eventually beta-2M, imaging also, obviously genetics, all combined will definitely increase our ability to stratify this stage of the disease course. As I alluded before, in this study, we also looked at immune profiles, and it was possible to develop an immune score relying on 2 different subsets of non-classical monocytes, activated cytotoxic NK cells, as well as a specific subset within stem central memory T cells that stratified patients into low and high risk of transformation according to low and high risk immune scores. And in this busy slide, what we are showing here is patients plotted according to the percentage of CTCs from low to high, and then the cutoff of 20 for the serum free light chain ratio, the cutoff of less or more than 2 grams per deciliter of the M protein, less or more than 20% bone marrow plasma cells, the 20 to 20 risk, the risk according to the new model based on CTCs, and the immune score, low or high risk. And the message that I would like to point out here is that, in my opinion, compared to tumor burden, genetics, imaging, etc., probably immune profiling would not be the most powerful risk factor to predict outcomes in smoldering myeloma. But I do believe that it will be important to identify outliers. And what I mean by outliers are, for example, patients that have all factors of low risk, but high immune score, high risk immune score, and eventually loss of immune surveillance and disease transformation. Or on the other side of the spectrum, patients with high CTC counts, having other risk factors, but eventually strong immune surveillance and without disease progression, even after 2 years or more of follow-up. As you may know, there is more and more expectation about what immune profiling can offer in this field, also because of the development of more and more immunotherapies that are showing remarkable efficacy in the treatment of myeloma. And I do believe that the potential is there. Probably immune profiling is not yet powered for implementation in routine clinical laboratories, but the potential is there. And I do believe that the potential is there because to some extent, immune monitoring could be uncovering immune suppressive states resulting from host, tumor, and treatment-related factors that, to some extent, will be more and more important, for example, regarding the risk of infection. And in fact, we do believe that immune monitoring or immune profiling could be helpful not only to predict survival outcomes in patients with myeloma, not only to eventually tailoring and defining optimal time points of vaccination. These are learnings from the COVID-19 era, also to improve monitoring of treatment efficacy beyond MRD, but also, and this is very recent data from our group, to predict the risk of severe infections. And this study, again, comes back from the COVID-19 era, but here to our French colleagues, I will in fact show the recent unpublished data in which we leveraged on all the immune profiling you can obtain using NGF, in other words, without extra cost, without extra complication in the laboratory. This is the same flow cytometry procedure that we typically do for the screening of plasma cell clonality, for MRD assessment, and this allows to measure easily 15 different cell types in the marrow, 10 in the peripheral blood, and as expected, there are significant differences across disease progression. For example, patients with relapsed refractory myeloma display further reduction of memory B cells and increased CD27 negative to positive T cell ratio, on top of other alterations probably related, for example, with increased inflammation in the bone marrow. And the reason why I emphasize those other cell types is because what we are seeing is that there are at least three immune biomarkers that can help identifying patients at higher risk of infection. And these are lower percentages of memory B cells, lower CD27 negative to positive T cell ratio, as well as a higher percentage of NK CD27 negative cells. And importantly, if we combine the three immune biomarkers into an immune score, you can see how the risk of infection increases over time in patients having none, one, or two or more immune risk features, and how this immune score is independent of disease stage in a multivariate analysis. And we believe that these findings that, again, can be easily obtained during routine procedures could be important, particularly now that anti-BCMA, anti-GPRC5D immunotherapies are being combined with other drugs, and the risk of infection could be even higher than what we are currently seeing in patients with myeloma treated with these drugs as monotherapy. Also important, in the 10 cell types that we can detect both in the marrow and in the peripheral blood, as you can see in the slide, there is a highly significant correlation in terms of percentage, and therefore these immune scores could be obtained either in the marrow, if a bone marrow aspirate is adequate for the staging of the patient at that given time point, or more conveniently in the peripheral blood in a minimally invasive manner. So I'll now talk about the use of big data. And one example I can share with you about the use of big data is how we generated the MGUS-like classifier. In other words, the possibility of using flow cytometry to identify patients with active myeloma, smoldering, or active myeloma having an MGUS-like infiltration in the marrow. And to this end, we leverage on flow cytometry and clinical data from more than 5,000 patients with different monoclonal gammopathies, MGUS, active myeloma, and there was validation in smoldering myeloma, a cohort of patients with myeloma from Czech Republic, and even in light chain amyloidosis. And if I remember well, in sake of time, I will not show the data on light chain amyloidosis, but there was clear reproducibility between the Spanish and the Italian cohorts. And we use all this data plus a logistic regression classifier based on two simple parameters, the percentage of bone marrow plasma cells and how much of those were clonal by flow cytometry to stratify patients into MGUS, intermediate, or myeloma-like. This is the application in smoldering myeloma. As you can see, those patients having MGUS-like disease according to flow, it's an MGUS-like behavior, definitely, and patients with myeloma-like disease have, I would say, ultra-high risk of transformation. And as you can see, there is reproducibility between the Spanish cohort and a cohort of patients studied in the University of Arkansas in the United States. And this is a sub-analysis. In fact, this was not published. This was presented at ASH, but it was not in the publication, if I remember well. But in the Spanish cohort, we looked at what would these phenotypes offer in terms of prognostication and patient management in terms of observation versus early treatment in two Spanish clinical trials that investigated early treatment in smoldering myeloma. These were the Kiridex trial many years ago, Lendex for 9 months, and then 2 more years with Len alone, if I remember well. And this was many years ago. And more recently, the Caesar trial for transplant-eligible smoldering myeloma patients that received a more intensive approach with KRD, followed by high-dose therapy, then KRD, consolidation, and maintenance with lenalidomide for 2 years. And these are the outcomes in terms of time to progression according to MGUS-like, intermediate, and MM-like. And these findings are already telling us that a few patients that were enrolled in the clinical trials, and these patients were enrolled in clinical trials at high-risk disease according to the Mayo or the older PETHEMA criteria, well, few had MGUS-like disease, meaning that this MGUS-like classifier is complementary to those other risk criteria, and there is no benefit on coming from early treatment. And in fact, if there is no risk of progression, obviously there is no benefit in intervention. In patients with MM-like disease, probably those with high risk or ultra-high risk of transformation, there is benefit, but probably the benefit is restricted to preventing symptoms, and probably early intervention is not curative, whereas probably in the intermediate subgroup, even though currently there is no evidence of benefit of early intervention, maybe with the adequate approach, this could be an area of high risk and hopefully high gain. Time will tell. Then in patients with active myeloma, obviously enrolled in the Spanish clinical trials, treated as active myeloma, you can see that these patients with MGUS-like disease by flow cytometry had unprecedented outcomes. You can see that the PFS and the OS rates at 10 years are close to 80%. And as you can see in the slide, these very promising outcomes. By the way, in clinical trials of fixed duration therapy, none of the trials was treatment until disease progression. These outcomes are independent of the depth of response, CR or MRD. And some may remember that, in fact, the whole purpose for us to develop the MGUS-like classifier was precisely to identify patients that are suboptimal responders, but nonetheless may enjoy long-term survival. And we believe this will be more and more important because, as you may know, there are now many ongoing clinical trials in which patients are being treated according to MRD, and there are trials intensifying or prolonging treatment based on persistent MRD. And what we show here is that there might be small subset of patients in which positive MRD is not associated with higher risk of progression, and therefore these patients should not be overtreated. And this is why it is important to identify biomarkers such as the MGUS-like phenotype. So now we are entering in the part of MRD in my presentation, and as you know, we are living a unique time in progress of myeloma therapy, with 19 drugs approved in the last 20 years, which obviously led to significant prolongation of survival outcomes. Fortunately, there are multiple new drugs and combinations to evaluate and hopefully to continue improving survival outcomes. Unfortunately, patients will have to wait for longer and longer periods for documented PFS benefit or a lack of survival benefit. And this was why it was so important to have the consensus on the value of MRD negative CR after 1 year after treatment initiation as an intermediate endpoint for accelerated approval of new treatments. MRD, as we know, was a strong candidate as an intermediate endpoint, and the reason is because if we look at the relative survival at 5 years based on the years of diagnosis throughout the evolution of myeloma treatment, and we look at the cumulative rates of undetectable MRD that have progressively increased with each new treatment option, I think it is unquestionable that the efficacy of new regimens, deeper and durable MRD responses, and prolonged survival are interconnected in myeloma. And because these are interconnected, this was the reason why we felt MRD could be an early endpoint. This was really big data. This was probably the largest data collection in myeloma to serve the purpose of having MRD accepted as an early endpoint within the I²2 team. And again, here we go. And again, the French group was a key partner in this consortium, providing a lot of MRD data in your IFM clinical trials. What we showed at the FDA almost 1 year ago and led to the approval of MRD negative CR as an early endpoint for accelerated drug approval in this disease was the fact that there is a very strong individual patient level correlation between MRD status and PFS as well as overall survival, and there was an encouraging trial level correlation between the treatment effects on the MRD negative rates and the treatment effects on PFS as well as OS. What does this mean? Well, this means that from now onwards, probably in a phase 3 randomized clinical trial, if there are differences between two arms in MRD negative rates, probably superior to 10-15% or 15-20%, this will probably be almost always associated with a meaningful improvement in PFS. Not any improvement in PFS, but a meaningful one. For example, a hazard ratio less than 0.60. In other words, a reduction in the risk of progression and or death of 40% or more. One recent example of this application was the Perseus trial. In the Perseus trial, the MRD negative rates measured after consolidation, in other words, 1 year after treatment initiation, clearly predicted what was seen 4 or 5 years after as the benefit in PFS on those patients treated with the quadruplet regimen compared to the triplet regimen, data VRD versus VRD. This said, it is important to recognize that in many studies, we see this phenomenon that despite the rates of MRD negativity are significantly higher with data VRD when compared to VRD, if the patient achieves MRD negativity, the PFS, definitely when it's defined with high sensitivity, 10 to the minus 6, the PFS is superimposable between the two arms, between the two treatments, more intensive, less intensive, 4 versus 3 drugs. And we believe this data is quite interesting because in a near future, when there are more and more options to treat newly diagnosed patients, the physician may well select one or the other option based on the expected tolerability of the patient to that regimen. For example, a patient that is more frail and potentially will tolerate less a 4-drug regimen could be selected for a 3-drug regimen based on the prediction that with those 3 drugs, the patient will anyhow achieve an MRD negative result. In other words, selecting treatment based on the prediction of what will be the MRD status with that treatment, and then confirm the prediction in a few months. And if the prediction was correct and the patient was MRD negative, then you know that PFS will probably be long. Now, a few years ago, we showed that in a proof of concept study that it was possible to predict MRD outcomes at diagnosis according to genetic, for example, deletion 17p and or 414 translocation, tumor biomarkers, plasma cell clonality in the marrow, and again, then we go again, circulating tumor cells, as well as immune biomarkers in the marrow that can be easily obtained, like I said before, with NGF. And using these biomarkers, it is possible to accurately predict what will be the patient MRD status in 70%, both transplant eligible and ineligible patients. And like the MGUS-like classifier, also the MRD predictor is available open access in the web pages that we created for these calculators. Finally, and towards the end of my presentation, I will show one example of pushing the limits of technology to improve patients' quality of life. And the example is about MRD detection in the peripheral blood. As you know, we rely mainly on flow cytometry, NGF, for the assessment of MRD, and the sensitivity of NGF ultimately depends on the number of cells you analyze. The more cells, the higher the limit of detection of the method. And the same applies for NGS. The more DNA, the higher the sensitivity of the method. Now, blood has 2 main advantages compared to the marrow. Well, 3. One is convenience for the patient, less invasive. Second is hemodilution. There is no hemodilution in the blood. There is no risk of hemodilution. It's blood. Third, you can ask for more volume. You cannot ask for a large volume of bone marrow because the more you ask, the more hemodilution. By contrast, in blood, you can potentially ask for as much volume as you need to increase the sensitivity of your method. And we thought, let's ask for 50 ml of blood, because in 50 ml, even in patients with cytopenias, we may end up analyzing systematically more than 200 million cells. And if we analyze more than 200 million cells, we could break the barrier of 10 to the minus 6 and enter into 10 to the minus 7 logarithmic range. But then we have a problem. And the problem is that in a routine clinical laboratory, you cannot analyze 50 ml of blood in a flow cytometer. You would collapse the laboratory. So you need to make it easier. And the idea we had to make it easier was to enrich those large volumes of blood into small aliquots of plasma cells using no more, no less than the typical CD138 beads we use for immunomagnetic enrichment of myeloma cells prior FISH cytogenetics. And using this strategy that we coined as blood flow, we showed that it is much more sensitive than NGF. It detects MRD in the blood, or we call it PRD, peripheral residual disease, twice as frequently when compared to NGF. And the reason is because almost always we are breaking the barrier of 10 to the minus 6. We are detecting PRD in the 10 to the minus 7, and even in some patients in the 10 to the minus 8 logarithmic range. And we showed, and this is a manuscript that is currently in review, we showed that detection of PRD is clearly prognostic in patients with active myeloma. The negative predictive value compared to the marrow is high, not perfect, but it's high. And as you can see in the slide, it is of independent prognostic value for PFS together with the MRD status in the marrow by NGF. And as expected, because it is more sensitive, it outperforms the PRD status according to NGF. Can we improve it? Yes. I mean, we cannot improve, at least not right now, the sensitivity of flow cytometry beyond 10 to the minus 7, but we can continue improving by adding more minimally invasive methods. And one candidate is mass spectrometry. And in this preliminary analysis of a larger project we have now ongoing, you can see in the slide that by combining blood flow with mass spectrometry, we can stratify patient risk according to PRD assessment, double negative, very favorable, promising outcomes. And if there is PRD detected either by one or the other method or the two methods, then the risk of progression is significantly higher. And as you can see in the slide, using the two methods, the negative predictive value was 84%. In other words, in 4 out of 5 times, every time we say no residual disease in the blood, there will be no residual disease in the marrow. By contrast, if it is positive, you know it will be positive in the marrow. You don't need to perform the bone marrow aspirate. And this is where we would like to go. Collect more and more data to make a reality what is currently a possible hypothetical algorithm of MRD and PRD assessment. During the first year of treatment, I believe that the marrow will remain as the gold standard specimen because the treatment is highly effective and the blood will probably be less informative. Then you may want to confirm the MRD status in the marrow every year, every 2 years, every 3 years, according to the patient needs and willingness for those consecutive bone marrow aspirates. And in the meanwhile, in order to avoid losing information about the MRD status, you could look into the peripheral blood and serum, for example, every 6 months, obviously without forgetting the added value of imaging modalities such as PET-CT. To conclude, I always say that there is no precision medicine without precision diagnostics, and I do believe that in addition to other techniques and features, flow cytometry analysis of tumor and immune cells is among that list that can help individualizing the management of patients with myeloma. I'll stop here, and I would like to thank you for your attention.
Thank you very much, Bruno, for this wonderful presentation. And we will have now the question and answer session that I said to all the participants to the seminars. You can ask your question in the Q&A section, and we will ask the question to Bruno, Jérôme, or myself. I have several questions, but maybe I will start with one already. Have you evaluate patients with extramedullary disease, and sometimes patients have a relapse out of bone marrow, and but because it's not always evaluating the blood, not evidence that there was a relapse because no sign of within bone marrow. And with your blood flow, are you sure that you will be able to identify those patients? Would it be useful in this situation?
I think it goes beyond that. In the let's say the original study we reported, it was published in January in JCO, where we used NGF, so not yet blood flow, NGF. We haven't found a specific correlation between the presence of extramedullary disease and detection of residual disease in the blood. So I think it probably goes beyond. Maybe if there is extramedullary disease, active, large, there will be cells in the blood, and both methods would be positive, PET-CT and NGF or blood flow, but we haven't found such an association. And with blood flow, neither. But also, for the time being, we are performing these studies in newly diagnosed patients. And as you know, the incidence of extramedullary disease in newly diagnosed patients is lower when compared to relapse refractory.
Thank you, Bruno. So,
okay, go ahead.
yeah, perhaps we could move to some questions that have been proposed. So the first one is, so Bruno, thank you for your presentation. With CD138 enrichment for blood flow, isn't it possible to make an adaptive biotechnologies MRD, which would be even more sensitive than flow cytometry?
Well, in my opinion, whenever you are comparing the two next generation methods, Euroflow and the adaptive, I think these are comparable. These are interchangeable and have the same performance. If you are comparing with a different flow cytometry method or a different sequencing method, then I don't know. But these two, I think they are very much similar. And yes, the approach can be used for NGS. And in fact, Benjamin Derman from the University of Chicago has already published in Blood Cancer Journal, I think at the end of last year, the use of immunomagnetic enrichment prior NGS in bone marrow aspirates. And like we did in blood, he also achieved 10 to the minus 7 with NGS. So the concepts are reproducible regardless of the next generation method you decide to use.
Thank you.
Next question.
Yeah.
Great talk. Many thanks, Bruno. Would combined ctDNA analysis help increase sensitivity?
Maybe. In that abstract we submitted to ASH in 2023, and probably towards the end of this year we'll try to finalize the study and report it. We performed a multimodal MRD approach using blood flow, mass spectrometry, and I showed one slide with that data, and we also used NGS, not focusing on the B-cell receptor, but rather on mutations. It was patient-specific. It reminds us of the ASO-PCR. It's more cumbersome, and the applicability may be reduced, as we remember from those old days, at least some of us. But the method was quite sensitive. In my opinion, the two methods that showed the more balanced negative and positive predictive values were blood flow and mass spec. But NGS was also sensitive and probably not as sensitive, but with the advantage of looking at mutations and having a better understanding of eventually genomic determinants of MRD resistance.
Maybe we can also talk about the cost, because I guess if you, I'm not involved in follow-up of patient at hospital, but I know you have to deal with the cost of all these approaches. Can you, what would you prefer, suggest to start with and to end with type of analysis depending on the result? For instance, if you have a lot of cells with flow cytometry, it's not necessary to go to mass spec and so on. Do you have some prioritization of your analysis?
You are alluding to MRD.
Yes.
Okay. Well, it's a nightmare. Regardless of what you do, it's a nightmare, and I'm afraid it will only get worse because of the political environment that we will not talk about. But I mean, if you need a result in a reasonable amount of time, in short amount of time, then in my opinion, flow cytometry, NGF, is the most cost-effective method because it's not that expensive. It's far less expensive than NGS, at least the commercial approach. And it's even less expensive than FISH, for example, at baseline, at least in our institution. So it will provide you a result in 24 hours at a reasonable cost. It is my understanding that mass spectrometry will be equally cost efficient. The cost is very reasonable, but it requires large numbers. You need to process a large amount of serum samples to make it cost efficient. So let's say in a mid-sized hospital in which you don't reach those numbers, you need to store in batch and then analyze all the serum samples, which means a delay in terms of time till having the results.
Thank you, Bruno. So just to move to another point, so I would have a question about the immune profiling and the risk of progression of smoldering myeloma. And just a question about what you observed with the immunophenotyping and what we could investigate as a marker that we could analyze in the plasma, in the sera. So could you find some link between immune profile alterations and inflammatory markers or something in the microenvironment? So do you think that the alteration of the immune cells is linked to the disease or is linked to the amount of tumor cells, or is it also linked to alteration of the microenvironment that could be related to inflammation or something like that?
I mean, the easy answer is yes. I think that for sure there is a connection. But, you know, and in fact, I was just looking at those results before connecting to the Red Talk, because, you know, in my opinion, whenever we compare, at least we using flow, for example, patients with myeloma, the immune composition in the marrow or in the blood of patients with myeloma versus healthy adults of the same age range, there are significant differences, but the magnitude is not dramatic. You don't see a completely different immune composition. So I think that a considerable fraction of immune alterations we see in patients with myeloma is coming from maladaptive aging. We don't know to quantify what exactly is coming from tumor, what is exactly is coming from age, but part, and probably a significant part, is coming from age. And because of that, I think that definitely looking into cytokines, proteins, either in the bone marrow supernatant or in plasma, in blood, could be complementary to looking into immune cell types. And even beyond that, I mean, looking to the function of those cells beyond the phenotype. I mean, T cells, looking at their T cell receptor, and if these react or not against myeloma antigens, there is much more to do. But obviously, the more you add, there you go, the cost will increase. And at least here, we always try to do research that could potentially end up in the clinical laboratory.
Thank you, Bruno.
So we have another question regarding the threshold of MRD. What is the difference, to your opinion, between 10 to the -5 or 10 to the -6?
Well, I mean, I will not answer on the specificity of the question. It's about how many cells above or below the threshold. The conceptual answer to the question is that, in my opinion, and not the concept, it's more the outcomes according to the treatment and disease. But differently from CLL, different from acute lymphoblastic leukemia, eventually AML, I'm not sure, but diseases where a 0.01% cutoff, in AML even 0.1%, has been used for treatment decisions with more or less efficacy. In myeloma, it does appear that any level of residual disease clearly impacts in survival outcomes. Even when we detect, and in fact, Aurore Perrault from the French group showcased this very well in 2018 in the Blood paper on the IFM 2009 study. Even when you detect MRD down to 10 to the -6, there will be reduced PFS and eventually OS. So, for example, for the MRD endpoint definition in the FDA and so on, -5 is probably the best cutoff because it's applicable across the board and will provide strong data in terms of comparing different regimens, etc. But for prognostication and eventually treatment decisions, we may well need 10 to the -6.
Thank you, Bruno. So another question from Bertrand. So it's interesting that immune monitoring in blood is somehow indicative of the immune tumor microenvironment niche in bone marrow. Have you found major differences according to responses?
It's an ongoing study. We do want to understand what immune profiling offers on top of depth of response in each treatment scenario and in each time point. It's a large endeavor. It's ongoing. Maybe at ASH, I don't know.
Yeah. And so Bertrand is also asking if single cell RNA-seq and this kind of characterization will be important to characterize the differences.
Definitely. To be honest, I think that currently most studies using single cell technology, I mean, flow is a single cell technology, but you know, I mean, single cell RNA-seq and so on, probably have not explored the full potential because in many studies it has been another way of counting different cell types. And for that, you have probably less expensive technologies. But you do see in some studies that, well, and there you go again, Ludovic from Toulouse has a very interesting study in NK cells published in Blood a few months ago. Anyhow, with these technologies, you may see an expansion of monocytes, and on top of that these are more pro-inflammatory. Or again, as you go for the T cells and their potential recognition of tumor cells. So obviously, these single cell technologies, spatial, another one, will offer much more. Let's see if the cost becomes more user-friendly.
I have another question regarding the immune control of myeloma cell, depending on the type of myeloma cells. Don't you think that, for instance, progression of patient with cyclin D1 deregulation is quite low, and maybe you're highly dependent on the immune suppression that increase, by contrast to patient with a 414 translocation with a very strong oncogene activation that can go on without waiting for immune dysfunction.
Maybe. I think that's an area that is, again, very poorly explored, the association between specific genetic alterations and the immune composition and the immune response. Maybe. I mean, we have the feeling, at least I do, but maybe I'm wrong. I'm the feeling that myeloma is not that immunogenic compared, for example, with solid tumors, but maybe there are differences. And by the way, as you see, I'm very French focused. You know, my second my hobby is my second work is to write commentaries to blood on French studies. There will be another one soon. and you know, the again, Aurore Perhoff, when she reported on behalf of the IFM, sorry, the IFM, yeah, the results from the MEDUS trial, the MRD negative rates are clearly different across genetic subgroups. And you know, you have isatuximab there, you have lenalidomide there, but who knows if these are related to different genetic, not only different genetic alterations, but also the immune profile of these patients. Time will tell.
But in your profiling of immune cells in your hands, did you look at the translocation of patient to see if there is some split or not of population?
We have the data. We have looked, but I don't have yet the results sound on the top of my head. But definitely the data is there.
Okay. And I had also another question. It is international recommendation on how you count your CTC. Is it an euro flow, international flow?
I mean, there isn't. We should work on it. And in fact, there is a very interesting consortium at European level doing a meta-analysis on CTCs, more than 2,000 patients, very robust data, probably will be presented at EHA. And I think that once a cutoff of CTCs is established for the staging of newly diagnosed myeloma, eventually also smoldering myeloma, we need to also provide consensus and guidance on how to measure. As you know, we always rely on EuroFlow, NGF, for the assessment of CTCs, MRD, also other immunophenotyping across malignant hematology. And we also need guidance on how to count. Counting, I think, is straightforward, as we do for other applications, but hopefully it will get there. It means that there is interest on the implementation of the CTC biomarker, and I think it's easy.
Because you have a clear cutoff, so it's important that all the laboratory use similar way to get a cutoff. Otherwise, it's difficult to compare.
My current understanding of the data is that regardless of the cutoff, it's a continuous variable, and with each log of increased CTCs, the worse the outcome. In fact, more than 0.1%, 1%, it's far, far below the criteria for plasma cell leukemia, but the clinical behavior is not that different from plasma cell leukemia. And regardless of whatever cutoff will emerge, in my opinion, whenever you detect above 0.01%, that is high risk.
Bruno, there is another question regarding the blood flow. So you use membrane markers only. You never check by performing permeabilization for CTC quantification.
We do.
Yeah,
I mean, because we stain for kappa and lambda.
Yeah.
It's intracytoplasmatic staining, so we do.
And how do you process with the enrichment with the CD138 bit? So how do you include that in the flow cytometry process? So can you keep the same time to provide the results, or?
Yeah, well, usually in 24, 48 hours. Obviously, it's more cumbersome in the laboratory. For a laboratory like ours, with both flow and cytogenetics, it's not that big of a deal, but obviously there is an additional step, that is the immunomagnetic enrichment. This said, after it's the same NGF protocol. It's 100 microliters of enriched plasma cells, and the time you lose with the enrichment, you'll gain in the flow cytometer because it's only a small aliquot. And the analysis, it's even easier because you remove a lot of nucleated cells, leukocytes, and I mean, it's not the same, but I always say it reminds us of, for those in flow cytometry, to some extent, the analysis of CNS lymphoma, looking at cerebrospinal fluid, for example. Low number of cells, but when present, easy to detect because you don't have background noise.
But you don't see any cells after you do your first enrichment.
No, you continue seeing because there is always impurity in
Yes.
enrichment, but we like it. I mean, we prioritize enrichment over purity because for flow you need your internal control of the other cells, so it's good to have impurity.
Of course. Another question. So cell enrichment techniques can improve sample purity, but is there a workaround for potential fast negatives or zero counts due to CTC clusters, epitope cloaking, barriers, etc.?
Well, I think this was answered before, right?
Yeah.
There is always impurity, but because you are doing flow afterwards, you can always check for each cell type that you are analyzing.
And false negative?
138 is not so important after for the identification of the plasma cells. So yeah.
No, and there is no, I mean, the clone is different,
Yeah.
and there is no full occupancy of the receptor. You can detect, you can measure the expression of CD38. 138 is reduced, but is still detectable.
Okay.
Okay.
Thank you. So, is
We
there
don't
other
have
question?
no more question in the chat. So, Jérôme, you close the
Yeah. So thank you very much, Bruno, for this wonderful presentation and all the things that you shared with us. Thank you, everybody, to be present and also to provide a lot of questions to discuss this interesting points with Bruno and all of us. And so now it's the end of the session. Thank you again to everybody, and thank you again for the organization, to Kalim, and also thank you again, Bruno, for the presentation and all the data that you shared with us.
Thank you for having me, and hope to see you soon.
Yes, of course, in Milan. We will see in Milan.
Yeah, if not sooner.
Okay.
Okay.
Thank you very much.
Thank you very much. Thank
Bye-bye.
you. Bye-bye.
Goodbye, Bruno.